Assalamualaikum and a very good day. My name is Madam Michelle Mariam binti Abu Bakar and I am from Biology Unit. Today we are going to learn Chapter 5, that is Expression of Biological Information, focusing on Subtopic 5.1, DNA and Genetic Information, and Subtopic 5.2, that is DNA Replication. Firstly, we are going to look at subtopic 5.1, that is DNA and genetic information. By the end of the lesson, you should be able to state the concept of central dogma. What is central dogma? Central dogma is the flow of the genetic information in the biological system. So the molecule that is involved in central dogma is DNA, RNA, and as well as protein. All living organism is made up of cell. Inside the nucleus of a eukaryotic cell, we can see there is a molecule that carries the genetic information of the cell known as DNA, where the genetic information it is carried in form of a gene. DNA is proven as a carrier of the genetic information from the experiment that is conducted by Griffith, Avery et al., and as well as Hershey and Chase. Now we are going to look at the concept of central dogma. The first flow of the genetic information can be seen during cell division. So the DNA, it is copied. This DNA, it will be copied by the process of DNA replication to produce two new DNA that is identical to the parental DNA. Besides that, DNA it carries the genetic information in form of a gene, which in each DNA it has many genes, and each gene it will encode to a specific polypeptide chain. So how does this occur? The DNA, which is the specific gene in the DNA, will undergo the process of transcription to produce a single-stranded mRNA strand, where this single-stranded mRNA strand will undergo the process of translation to produce a polypeptide chain. The concept of one gene encodes for one protein was proposed by the experiment conducted by Biddle and Tatum in 1941, which then it was refined by other scientists, which concludes that one specific gene it encodes for one specific polypeptide chain. In another case, the flow of the genetic information can be seen in virus, which is a special case. There are viruses which the genetic information is RNA. Example is coronavirus. So the RNA molecule, which is inside the virus, it will undergo the process of reverse transcription to produce the double-stranded DNA. Next, we are going to look at the next subtopic, that is DNA replication. In this subtopic, we are going to look at how to explain semi-conservative replication of DNA and explain the enzyme and proteins involved in DNA replication. What is DNA replication? DNA replication is the process where two new identical DNA is copied from original DNA. It can occur in a nucleus of a eukaryotic cell, or also it can occur in the cytoplasm of a prokaryotic cell. There are three alternative models for DNA replication. The first one is semi-conservative model. In semi-conservative model, the parental DNA strand, it will be separated, which each DNA strand, it will act as a template to synthesize a new complementary DNA strand to the template. So the old DNA strand is the purple colored DNA strand. The newly synthesized DNA strand is the blue color. So now I would like you to figure out how does the second replication occur to produce this kind of result. In conservative model, the parental DNA strand will reassociate after they act as a template for the new DNA strand to form. Thus, it will restore the parental double helix. So each DNA molecule, it consists of 2 old strands and 2 new strands. The last alternative model for DNA replication is dispersive model, which each strand of a daughter DNA molecule, it consists of a mixture of the old and newly synthesized DNA. So which DNA replication is the correct one? So the DNA replication was proven to be semi-conservative by the experiment that is conducted by Matheson and Stahl. Before we look at the model of the DNA replication, let's look at the DNA molecule. DNA molecule consists of 2 polynucleotide strands, and each strand it is complementary to the other strand by the nitrogenous base. The base pairing rule is where adenine it pairs up with thymine by 2 hydrogen bond, and cytosine it pairs up with guanine by 3 hydrogen bond. Now we are going to look at the semi-conservative model for the DNA replication. In the first step, the 2 DNA strand it will be separated, which each parental DNA strand now it will act as a template to produce new complementary DNA strand according to base pairing rules. So dark blue color DNA strand, which is the template, is the parental DNA strand, and the light blue color DNA strand is the newly synthesized DNA strand complementary to the template. 2 new identical DNA strands now are formed, where each dotted DNA consists of 1 parental DNA strand, the dark blue color DNA strand, and also 1 newly synthesized DNA strand, which is the light blue color, and it is complementary to the template. After we have already looked at how DNA replication occurs by semi-conservative, now we are going to look at the next learning outcome, that is to state the enzyme and protein involved in DNA replication and describe the mechanism of DNA replication and the enzyme involved. There are 5 different enzymes and protein involved, that is helicase, single-stranded binding protein, topoisomerase, PRIMASE. There are 2 types of DNA polymerase, that is 1 and 3, and DNA ligase. The first enzyme involved in DNA replication is helicase. Helicase is the enzyme that catalyzes the unwinding of the DNA double helix, and it will separate the 2 parental DNA strands to become single-stranded DNA. After helicase has done its job, then the single-stranded binding protein are the protein that will hold the separated DNA strand apart while each DNA strand acts as a template. Unwinding of the DNA double helix by helicase, it will create strain to the DNA double helix ahead from the unwinding. To relieve the strain that is caused by unwinding, it is catalyzed by the enzyme called topoisomerase. PRIMASE is the enzyme that catalyzes the addition of RNA nucleotides that is complementary to the DNA template from 5' to 3', which this short segment of RNA nucleotides, it is known as RNA primer. There are 2 types of DNA polymerase involved in DNA replication, that is DNA polymerase 3 and DNA polymerase 1. DNA polymerase 3 catalyzes the addition of free DNA nucleotides to the exposed bases on the DNA template by the specific base pairing, at the 3' end of RNA primer. DNA polymerase 1, it will catalyze the removal of the RNA primer and replace it with the DNA nucleotides. Last one is DNA ligase. So DNA ligase are the enzyme that is catalyze the joining of the sugar phosphate backbone of all Okazaki fragments. So they will join the Okazaki fragments by forming phosphodiester bond. After we have already learned about the enzyme that is involved in DNA replication, so to see how DNA replication occurs, please scan the QR code to watch the video. Before we end our class, we can do some quiz to see if you understand the topic. Thank you, and we'll see you again in the next subtopic.